Hello book lovers, this is Vanapalli Harshiki, you are seeing Family Notes. Today our topic is clotting factors. Clotting factors is one of the most important topic of this chapter. Let's start our topic. We have 12 factors, that is factor 1, 2, 3, 4, 5, 7, 8, 9, 10, 11, 12, 13. We don't have the sixth factor. Factor 1 is known as fibrinogen. Factor 2 is prothrombin. Factor 3 is tissue factor or thromboplastin. Factor 4 is calcium. Factor 5 is labile factor or also known as proaxilrin or AC globulin. Factor 7 is a stable factor or proconvertin. Factor 8 is antihemophilic globulin or antihemophilic factor A. Factor 9 is Christmas factor. It is easy to remember and it is also called as plasma thromboplastin component and antihemophilic factor B. Factor 10 is known as Stewart-Prower factor. Factor 11 is known as plasma thromboplastin antecedent or antihemophilic factor C. And coming to factor number 12, it is known as Hageman factor and 13th factor is known as fibrin stabilizing factor. There is no sixth factor. These clotting factors combinedly will cause the clotting of the blood so that causes hemostasis. Now let us know about hemostasis. The process of blood clotting and then the subsequent dissolution of the clot following repair of the injured tissue is termed as hemostasis. Hemostasis comprises of 4 major events that occur in a set order following the loss of vascular integrity. Those 4 events are vasoconstriction, platelet plug formation, coagulation that is blood clotting, and fourth one, fibrinolysis. The initial phase of the process is vascular constriction. This limits the flow of blood to the area of injury. Next, platelets become activated by thrombin and get aggregated at the site of injury, forming a temporary loose platelet plug. The adherent platelets clump to each other and release certain substances including ADP, which attract more platelets to the site. Passing platelets stick to those already at the damaged vessel and they too release their chemicals. This is a positive feedback system by which many platelets rapidly arrive at the site of vascular damage and quickly form a temporary seal, the platelet plug. This platelet plug formation is usually complete within 6 minutes of injury. And next, coming to the third event, that is to ensure stability of the initially loose platelet plug, a fibrin mesh forms and entraps the plug. If the plug contains only platelets, it is termed as white thrombus. If red blood cells are present in it, it is called as red thrombus. The coagulation of blood will be achieved by 2 pathways, that is extrinsic pathway and intrinsic pathway. We will see about these extrinsic pathway and intrinsic pathway in the next video. Now let us continue with our topic. As the clot is formed, that clot shrinks to pull the edges of the damaged vessels together and reduces the blood loss and closing of the hole in the vessel wall. Now coming to the fourth event, that is fibrinolysis. After the clot has formed, the process of removing it and healing the damaged blood vessel begins. That means the breakdown of the clot or fibrinolysis is the first stage and plasminogen trapped within the clot as it forms, it converted to the enzyme plasmin by activators released from the damaged endothelial cells. This plasmin breaks down fibrin to soluble products that are treated as waste material and removed by phagocytosis. As the clot is removed, the healing process restores the integrity of the blood vessel. Now coming to the control of coagulation. The process of blood clotting relies heavily on several self-perpetuating processes. That is, once started, a positive feedback mechanism promotes their continuation. For example, thrombin is a powerful stimulator of its own production. The body therefore possesses several mechanisms to control and limit the coagulation cascade. Otherwise, once started, the clotting process would spread throughout the circulatory system instead of being limited to the local area where it is needed. The main controls include the perfect smoothness of normal blood vessel lining prevents platelet adhesion in healthy undamaged blood vessels. And second one, activated clotting factors remain active for only a short time because they are inhibited by natural anticoagulants such as heparin and antithrombin 3, which interrupt the clotting cascade. Here, the natural anticoagulants like heparin and antithrombin 3 will inhibit the action of the clotting factors within a very short time of the clotting. If you like the video, please subscribe our channel.